Good evening, everybody. Welcome to BrisScience. Thank you so much for coming out on this blustery winter's night. My name is Joel Gilmore. I'm your MC for this evening, and this is, of course, BrisScience, hosted by the University of Queensland. It is a free public lecture series of science held once a month here at The Edge, the fantastic Edge at the State Library of Queensland, our wonderful venue partners. Before we start, I would like to acknowledge the traditional owners of the land on which we're meeting tonight and to pay my respect to elders both past and present. I'd also like to acknowledge those whose ongoing efforts to protect and promote Aboriginal and Torres Strait Islander culture leaves a lasting legacy for future elders and leaders. Tonight, we have a wonderful speaker from UQ. We're going to talk about plants and medicine. We'll get to that in just a moment. A few bits of housekeeping first that most of you will know. But if you're new to BrisScience, of course, the first thing is make sure your phones are on silent. We don't need to phone any friends tonight. But you can keep Twitter loaded up because we will be taking questions and comments over Twitter throughout the night, and we will be live tweeting as well if you need to look up a particularly interesting chart later on before the videos go up. So you can tweet us with hashtag BrisScience, or you will have picked up a question sheet on the way in. You can write down your question, and at the end of the talk, we'll go around and collect them and go through as many questions as we can at the end of the night. We'll also be having some food and drink afterwards, so we'd love for you to stick around, have a chat with yourselves and also with our fantastic speaker. I think that's all of the housekeeping. So tonight, it's my great pleasure to be introducing Bronwyn Smithies, who is from the Institute of Molecular Biosciences at the University of Queensland. And Bronwyn is interested in how plants work and how they can be used to benefit society in new and interesting ways. Which means if you weren't excited about plants before, don't worry, they will grow on you. Thank you. All right, we'll work from there, great. Tonight, Bronwyn is going to be talking about her research into how nature's amazing laboratories, plants, might be able to be used to produce new medicines more cheaply and more efficiently than existing techniques. So to tell us more, please put your hands together and welcome Bronwyn Smithies.
I have got my phone with me, but I'm not going to be checking Twitter. It's just so I can check the time. So thanks so much for having me. I'm Bronwyn, and I am going to tell you about growing medicine. And hopefully I can tell you a little bit about what that means for me and my research group at UQ. And also a little bit about the global kind of situation at the moment and some research going on worldwide. So getting straight into it. I just wanted to start with this photo. I took this photo because I'm from Tasmania, and before I moved to Brisbane for my PhD, I was driving past these gorgeous poppy fields, and I sort of put it up here not only to show you that Tasmania is very pretty. If you haven't been there, you should go, but also just to kind of give you a picture of what it might look like to grow medicines in plants. I'm not going to be talking about poppies today, but this is an example of a highly regulated and controlled medicinal crop. So Tasmania makes a lot of the world's morphine and codeine from these opium poppies. And so this might be what some of our future work could look like in years to come. So I'm going to start with the question that I always get when I meet people, which is, what are you doing for your PhD? So I'm actually studying for my PhD, which means I'm doing a big 3-year project, and then I'm going to write a thesis at the end. So first of all, thanks for having me so I can have a bit of a break from writing my thesis tonight. So when I tell people what I do for my PhD, I usually say, well, I make drugs in plants, or I make medicine in plants, and I get 2 main responses. So the first one is, a lot of people think, oh, that's really nice, like traditional medicine, herbal medicine, oh, that's really nice that we're looking into that again. And I do have to say that although we're working with that idea, this is actually, my work has a bit of a modern twist on this. So we're actually getting plants to make some medicines that they couldn't make before. But I'm going to explain that to you tonight. And of course, the second response that I usually get when I say I make drugs in plants is this one. So I am just going to make it clear that actually I'm not researching medicinal marijuana. So sorry if that was why you came along tonight, but that's not what I'll be talking about. So actually tonight I'm going to talk about the research that's going on in my lab at the IMB, at the Institute for Molecular Bioscience. And I'm going to take you through this story of discovering drugs in plants. We have these little molecules here that I'm going to explain what they are soon. How we're changing those into new drugs that we can use to treat different diseases. And then why we want to make our new and improved versions of these molecules in plants. So first of all, I wanted to get people to think maybe about how your medicine is made. So it might not be something that you think about when you go and buy some tablets from the pharmacy, but if you think about it, you can probably imagine that a lot of your pharmaceuticals come from a laboratory somewhere, perhaps a factory full of really big laboratories. And so I was looking for some pictures of some of these labs, and I found a video from a big pharmaceutical company that has very big production facilities. And I just took a few screenshots from one of their videos of their facility. And as you can see, it's all shiny and expensive looking. There's engineers and there's scientists, and they have these huge, big, shiny facilities for making pharmaceuticals. And actually, at this particular company, all their staff have free healthcare, and they can even go and do yoga classes if they want to, yoga and Pilates, so they're well looked after. And I look at this, and maybe you look at it as well and think, oh, like, this looks really nice. And I'm pretty confident that when I go to the pharmacy to buy my medication, I know it's coming from somewhere that's regulated and it's clean, and I know that it's going to be safe. But the other thing that I see when I look at these pictures is perhaps, wow, this looks really expensive. And the problem is that there are 2 billion people worldwide who do not have access to essential medicines. And cost is a huge factor in prohibiting them from access to that medicine. So the World Health Organization, this is a bit old, there is a new Director General now, it's a couple of years old. But basically there was a report about access to essential medicines, and they said that the number one cost for medicines for developing countries is paying staff and training staff, and the second biggest cost is drug procurement, so actually getting those medicines. And for a low to middle income country, they are spending 20 to 60% of their health budget on medicines, whereas a developed country like Australia would spend less than 20%. So there's some inequality here. And although it's a big, complex issue, we would like to think that perhaps we can try and do our small bit to try and improve this situation. And we think making medicines in plants could help reduce the cost of the manufacture of these, or of any medicine that we could potentially make in plants. So some of the benefits of plant-based production of medicines include perhaps that the facilities that we need to build to grow plants are going to be cheaper than perhaps those expensive ones that I showed on the previous slide. Also, the technical input. So it probably wouldn't cost as much to train a workforce with basic agricultural skills to work in one of these plant factories. And also scalability. You can imagine that growing a lot more plants or a lot less plants would be easier to set up and manage than perhaps expanding your facility or downgrading it. And I'm not going to go through everything on this table, but this is from a publication just outlining some advantages of plant-based production. So we've got plants at the bottom here. And it's just saying the cost is very low, scale-up capacity, as I mentioned. We have things like a low risk of contamination of these facilities. It's inexpensive to store these products. The only disadvantage really here is that the production timescale can be a little bit long. But I'm going to give some examples of actually how some companies are overcoming this so they can make new medicines in plants very, very quickly, in a matter of weeks, actually. So at the moment, this is a big global effort. There is, if you are a researcher like me or my colleagues, you can become part of a scientific organisation. And there is one called the International Society for Plant Molecular Farming, which just means making medicine in plants. And currently there are people from 21 countries in this organisation. And I'll just give a couple of examples of what's going on in the world right now. So there is a company in Canada called Medicago. And right now they are making influenza vaccines in plants. And this is straight from their website. So this is actually what their facility looks like a big green factory. And so their influenza vaccine, their flu vaccine, they can make it in a matter of weeks. So if a new strain of the flu comes out, they can quickly identify that and get the plants making the vaccine. Another fun example is in Japan. There is a company making a product, actually it's a veterinary product for dogs for gum disease, and they're making that in strawberries, which is kind of nice. And there's also another company in the States that was involved in developing a vaccine for Ebola back during a couple of years ago during the Ebola epidemic. And they were able to test that on compassionate grounds, and now they've taken that through to phase 2 clinical trials, I believe. So there's a lot going on, and it's very exciting. But now I'm going to get to the research that's going on here, because that's what I know and that's what I do. So I am from Professor David Craik's laboratory at the Institute for Molecular Bioscience, and it's quite a big group. There's about 50 of us, which is quite big for a research group. And that's because we have people doing a whole range of things. So it's not just the plant researchers like me, but we have chemists and biologists and people working with cancer cells, and everyone is working towards developing new medicines. And then this group of people over here, this is the fun bunch of people that I get to work with most of the time. We are the people who are going to try and make these new medicines in plants. So our research is about a particular type of molecule. So I'm just going to explain exactly what we're looking at. So we look at peptides, and some of you may or may not know, peptides are actually just small proteins. So what is a protein? If you think about protein right now, you're probably thinking about food, and maybe you're thinking about a steak or a bowl of tofu. And you're right, so we eat protein from our diet. And proteins are actually just a big chain of amino acids. So amino acids are these small molecules that can link together in a big long chain, and there can be thousands of them, and that's what makes a protein. And proteins are found in all living things and in every cell in your body. So they help the cells in your eye, they help you to see. The proteins in your stomach help you digest food, and there are proteins that hold everything in your body together. And there are also proteins in all other living things. So this jellyfish here can glow in the dark because of a protein, and these plants here can harvest energy from sunlight because of proteins in the leaves. And what we're working with are peptides, which is just a name for cute little proteins. So that means that they have 50 amino acids or less, which is quite small compared to some proteins that have thousands of amino acids. Some examples that you might know include insulin. So we all know that insulin helps us manage glucose in our body, and if we have problems with the insulin production in our body, we can get diabetes. And insulin is actually just a little peptide. Actually, it's two peptides joined together. You can see the blue and green there. So this is what this peptide would look like if we could see it. But obviously, it's very small. And another example you might have heard of is oxytocin. Oxytocin is also known as the love hormone, and recently mentioned on The Bachelor. I don't know if there are many Bachelor watchers here. There's one. So oxytocin is the hormone that allows you to feel good, like when you're falling in love, and it's so cute that it even makes you feel so strongly about your loved one that you're going to go out and buy a necklace of the chemical structure of oxytocin. So some peptides are cute, and some are less cute. So when you mention peptides, a lot of people's minds go straight to perhaps the misuse of peptides, particularly in the sporting industry. I don't know if you think that's cute, but regardless, the point is that peptides can be active inside our body in a lot of different ways. And so we want to use that activity to be able to treat different diseases. So peptides as medicines have a few advantages that are really, really positive. So they can have high activity. That means you can take less and get more activity. They're highly specific, which means they're going to have less side effects. Low toxicity, they don't accumulate in your body, and they're hugely diverse. But there are a couple of reasons that peptides haven't taken over the pharmaceutical market. They're out there, but they're not the main drugs out there. And that's because a lot of peptides break down very easily. So your body thinks they're food and it just digests them. And also they can be expensive to produce. But in our research, we're looking at addressing these problems. And in particular, the cost of production we would like to address by making these implants. But I'm going to go through a little bit about the specific peptides that we look at and what we're trying to do. So peptides as medicines, we think that the potential is there, but there are these downsides, and we want to try and address those. So the big disadvantage for peptides is that they break down in your body. And I mentioned that peptides are made of amino acids. This is just a list of all the amino acids that your body needs. And some of them might be familiar, or they might all look like something you've never heard of. But the point is, we need all of these amino acids, but our body can't make them. We can make some, but we need to get some from our diet. And so that's why, we need to get these ones from our diet. And so that's why our body thinks that proteins and peptides are food, because we have enzymes in our tummy that will come along and chomp up any kind of protein or peptide that we eat, and they're going to break it down into the individual amino acids, and then our body can use those to rebuild other proteins that we need. So how can we get around this? We think that if these enzymes are attacking the ends of the peptide, that if we could block the ends, then perhaps the enzymes won't be able to break it down. So this is our great plan. Now he doesn't know what to do. And we call this, now we call this a cyclic peptide. So we call it cyclic because if you unraveled it, it would just be like a circle with no ends. And actually, this isn't our idea. So nature kind of invented this before us, and we find cyclic peptides occurring naturally in plants and animals and bacteria. And the particular cyclic peptides that I'm going to talk to you about today are called cyclitides, which makes sense. It sounds like cyclic peptide. So there are different types of cyclic peptides, but all the ones that we call cyclitides all have the same structure. So this kind of looks like a chain in a circle. If each one of these little circles is an amino acid, every single cyclitide has 6 cysteines. So cysteine is one of those amino acids. And the great thing about cysteines is that they can bond together. So if these lines are like a chemical bond, every single cyclitide has 6 of those, and that's how we know it's a cyclitide. And cyclitides, we have found these in 5 plant families. So not all plants can make these. But, for example, every plant that we've looked at in the violet family, the Violaceae, every plant in that family can make cyclitides. Whereas some of these other families, the Petunia here from the Solanaceae, we have Fabaceae, which is the bean family. This one here is gac fruit from the curcubit family, and up the top we have the rubiaceae. So some plants in those families can make cyclotides and others can't. And although this is kind of a cartoon version of a cyclotide, it does have a 3D structure. So this circle can fold up and then it will look something like this. You can see these yellow lines here are these bonds here. And so we're interested in finding cyclotides from nature. We also look at some other cyclic peptides that they're not technically cyclotides, because you can see this one has 2 cysteines instead of 6. And this one is another little funny one called an orbitide, and I won't be talking about those. But just to give you an idea that there are different types of cyclic peptides out there. So in our group, we're looking at everything that you could possibly think of to do with cyclotides, including their evolution, how they're distributed in those plant families, discovering new cyclotides, their 3-dimensional structure, as I mentioned, their function. So we think that plants make cyclotides to protect themselves against insects. We're looking at applications in agriculture, in the pharmaceutical industry. We're looking at how they work and how to make these synthetically. So that means how do we make them in the lab, and also how we make them in plants, which is obviously what I'm here to talk to you about. So I'm going to talk to you about the plant-based production and the pharmaceutical application of these cyclic peptides. So keeping on the theme of medications from plants, cyclotides were discovered in the 1970s. A doctor was in the Democratic Republic of the Congo and noticed that women were drinking a tea that was being made from this plant here, which the locals called Calata calata. The Latin name is Aldenlandia affinis. And they were drinking this tea to aid with childbirth, so it was actually accelerating childbirth for them. And they thought, if there's something in this tea from this plant that can survive, first of all, being boiled, and then also being drank and ingested, and it's still not being broken down in their tummy, then it must be a pretty stable peptide or something. They didn't know it was peptide at the time. But you can already tell that it's starting to sound like perhaps it was one of our cyclotides. But it wasn't until the 1990s when it was found that it was actually a cyclic peptide. And then David Craig, who's the head of my lab, determined the 3-dimensional structure. This is the picture I put up before. So this means how it's folded together. And he and his colleagues called these cyclotides, and that's when the word came about. And this kind of goes back to the idea of traditional medicine. But actually, since the 90s and since the discovery of cyclotides, we've put a modern twist on this, because we're actually redesigning these molecules. So a bit more about cyclotides. They're incredibly diverse. We think there's tens of thousands of cyclotides out there, and we've already discovered several hundred. And they're diverse because of how many there are, but also because of their structure. So that way that they fold up, that can happen in 3 different ways. And cyclotides can also be produced in different parts of the plant, and also some plants can make many, many cyclotides. So here we've got the first prototypic cyclotide. This is from that plant that was called Calata calata, and it folds up into this 3D shape. But we also have heaps of other cyclotides, and for example, we love this plant. It's called Clitoria ternatea, or butterfly pea. That's PEA. A lot of people, if you just say butterfly pea, they think you're talking about something else. So butterfly pea can make cyclotides in the shoot. And here's an example of the sequence in the flower. So you can see here, we're replacing some of these amino acids with different ones. In the leaf, in the roots, and in the seeds of the plant. And so every time we have a new cyclotide, some of these amino acids are a bit different. But the 6 cysteines that I mentioned always stay the same. And so all these different cyclotides have some natural properties, such as insecticidal. So there is a product now on the market. There's a company that we were working with developing an insecticide to protect crops. And that's an all-natural organic insecticide called Zero-X. And there are some other activities that have been reported. And when I say activity, I mean it can have some kind of effect. So the uterotonic here, that was the first example of the aiding in childbirth. We've also got anti-HIV and anti-cancer. But we think, since there are so many cyclotides out there and the amino acids can vary so much, maybe we could design our own. And that's where we get to the idea of actually introducing properties into cyclotides. So we call this idea grafting. And if anyone here is a gardener or a horticulturalist, you might know grafting is usually used to explain putting 2 plants together, so perhaps the roots from one plant and the shoot from another plant. And this is done in industries like the grape industry, where they choose roots from a grapevine that can resist pests in the soil or are very drought tolerant. And then they choose the top part of the plant, the shoot, which might make a lot of grapes or be very productive. And they graft those 2 together. And we're trying to do that as well. So we're taking a cyclotide that had certain properties, and then another small peptide that has a property that we like it can kill cancer cells, and we're joining those together. So we're sort of up to here in the story. So we've discovered cyclotides, and now we want to chop and change some of the amino acids intentionally so that we can treat different diseases using these cyclic peptides. So we're going to take a small peptide that we know is active against cancer or HIV, and we're going to stabilize it, because as it is, it's not very stable, and that enzyme can come along and chomp on the ends. So we're going to stabilize it by putting it inside the cyclotide scaffold. So this is a big, long list of a lot of examples that have been looked at by us and other researchers around the world. And it's just showing, sorry, now I'm pointing down here instead of pointing up here. It's just showing a big list of all the different diseases that we've looked at targeting using grafted cyclotides. Now this is actually, a lot of this research was done and published, and that means that the publications are out there with all the supporting evidence. I'm not going to go through them all, but these are references. If you need them, I can also show them to you later. But so far, we have designed and tested all these new cyclotides, and we've done it all in the lab. So when we're testing our new cyclotide against breast cancer or leukemia, we actually made the peptide in the lab, which means we synthesized it. But what we would like to do is actually make them in plants. So we get to the case for cyclotide production in plants. And I'm just going to check the time. Good. Okay, so hopefully this is where everything is going to come together in terms of what I've talked about so far. So what we already know is that there are advantages to plant-based production of pharmaceuticals in general, in terms of the cost and the safety and the quality. And what we also know is that plants know how to make cyclotides. So we discovered them in plants. We know that plants know how to make that special cyclic structure. And although we've made some changes, we think that the plants might still know how to make our new cyclotides. We also know that plants can make cyclotides in different parts of the plant. So that's nice for us. If we, say, want to make a cyclotide in the fruit or just in the leaves, we can try and do that. The other thing that I haven't mentioned is that making cyclotides synthetically, so making them in our lab, is quite expensive, And it does produce chemical waste. So if we're making we only need a tiny bit for our experiments. But if we're talking about scaling up, so commercial production or getting enough for a big clinical trial, then we would have to make a lot of this stuff. And there was a recent article that suggested that for 1 kilogram of peptide produced, there's multiple tons of chemical waste. So it's not ideal, and that's another reason why we would like to make our cyclic peptides implants. And there are people looking at, even if we make these implants, how can we deal with the plant waste? So that's what's highlighted. There was a study about the side stream revenue, so we think that it's a win-win situation. So we have this really pretty facility called the Clive and Vera Ramaciotti facility, and this is part of our lab. And so a few years ago, David received a grant from this Clive and Vera Ramaciotti Foundation to set up this growth facility for us. So this picture sort of looks a bit like it's been coloured wrong, but actually it is actually pink inside this room because we have lights that are specific for the plants. So plants don't need all the same wavelengths of light that we need to grow. So we can optimise the light colours that they need. We can have LED lights that are much more efficient than regular white lights. And we have this great team of people. And so what we're trying to do is turn a plant into a biofactory. And it's all well and good for me to stand up here and say that that's what we're doing, but it's kind of hard to imagine how that might work. So hopefully you can visualize maybe what it might look like. So a plant can make a cyclotide. That means that it has a gene that has a piece of DNA that has a blueprint that tells that plant how to make, in this case, the cyclotide. And so what we can do is design our grafted cyclotides. We pick one that we go, hey, this is going to be a good drug lead. And then we need to modify the gene in the plant to tell it how to make our different new cyclotide. And so this is where we put the gene into a new plant. And we hope that the plants will like this and make the cyclotides for us. So we're hoping to do this for a lot of our cyclotides, and as the table before showed, there are a number of diseases that we're looking at targeting. So now I'm going to introduce some of the key players in my research in particular. So the first one here is called Nicosia benthamiana, and you might realise that Nicosia sounds a bit like nicotine, and actually this plant is a relative of tobacco, but it's not the smoking tobacco. It's actually an Australian native relative of tobacco. So we use this plant to produce our cyclotides because it grows very quickly, and we can get our product out of the plant within a few weeks. And it grows really quickly and it makes all these leaves, so we've got a lot of material to work with. And this plant is actually the same one that's being used to make the influenza vaccine that I mentioned at the start. So we also have this other plant called Arabidopsis Thaliana, which looks like a bit of a weed. And if you work with plants at all, like as a researcher, you know this plant because every single lab in the world that does plant research has Arabidopsis Thaliana growing in there, because it's a bit like the lab rat of the plant world. It's the model species. So although it doesn't look very exciting, it's really, really great for us because there's a whole international community that are experts on it. It is also quite fast, not as fast as the benthamiana, but in a few months we can look and see if it's making the cyclotides that we want. And also make lots of seeds, although they're really, really small. So this is just 100 seeds actually, and it's smaller than the top of a pen. But that's good because I can store a lot and I can take up all the storage space with lots and lots of seeds. So very quickly, I also just wanted to explain how we do this, because it's a question that I get a lot. So how do you actually get a gene into a plant? And the way that we do this, there are a few different ways, but the way that we do this is that we use a type of bacteria. It's called agrobacteria, and it lives in the soil, and it actually can infect plants just naturally. It actually causes crown gall disease, which is a disease that affects some crops. They get kind of knobbly looking growths. But the point is that when it infects a plant, it actually puts a little bit of its own DNA into the plant. And so what we can do in the lab is engineer this bacteria to have the gene that we want in it. Then we can infect the plants with that, and it will hopefully put that gene that we want into the plant. So we're kind of using this nifty trick from nature to do the work for us. Funnily enough, it's actually, as I said, it's naturally occurring bacteria, and it doesn't normally cause permanent changes to the genome of the plants that it infects. But there's actually one case where back in evolutionary history it did, and that is the sweet potato. So all the sweet potato that we eat today actually has some genes in it from agrobacteria. So thousands and thousands of years ago, the bacteria put a little bit of its own genome into the sweet potato genome. So it's almost like a natural GM plant, if you like, without getting too controversial. So when we use this bacteria to infect the plants, we can have transient expression of our cyclotides. And all that means is that the plant is just going to make it for a few days, up to 7 days, and then it's going to stop. And it means that the DNA that we put in is not permanently incorporated into that plant, and its offspring would not be making our cyclotides. So after we're done with them, we throw them in the bin. Or we can have stable expression, which means that we have permanently put a gene into the genome of that plant, and the seeds that we get from that plant will also be able to make cyclotides. So there are cases where we want one or the other. There are advantages to both. In our pretty pink room, we also grow some other plants. These are some of the plants we grow that naturally make cyclotides, and we use those to study how plants actually do this, how they put the cyclotide together. But for the most part, we're making cyclotides in these plants because they don't actually make cyclotides already, and it means we won't get interference from the native cyclotides. If we have an extract with cyclotides in it, we know what cyclotides are there. So back to our big list of designer cyclotides, I'm just going to give a couple of examples of some of the designer grafted cyclotides that we're trying to make in our plants. And the first one that I'm going to talk about is a drug lead for chronic myeloid leukemia. So leukemia, as you might know, is a blood cancer, and it's caused by a big protein, again the protein, that is actually caused by your body makes a bit of a mistake. There are two proteins that are normally there in the cell, but it kind of chops them in half and puts half of this one with half of this one, and then you have this fusion protein that's actually a problem and not acting the way it should. So what we want to try and do is look at that big fusion protein that's the problem and try and stop it from working and causing the cancer. And there are drugs on the market that are very effective for chronic myeloid leukemia because they stop this big fusion protein from working. But the problem is that some of them, people can develop resistance, so then that drug doesn't work for them anymore. There are new drugs on the market, so people can take those new ones, but the risk is that they're also going to develop resistance to those drugs as well. And the other thing about these drugs, one's called imatinib, one's called ponatinib. They're very expensive. And so what we would like to do is use our cyclotides to, here's one here, to create a new drug that is less likely to have, less likely for people to develop resistance and is cheaper. So this is where we're getting a bit detailed and intense. But all this is showing is that there's a lot of sequences here of these amino acids, And some really clever people in my lab have gone through it and tested every single one of those against that big problem protein for leukemia, and they've tested them and tried to find out which ones are actually going to work. And we also have very talented people in our lab who can make computer models of these molecules. So this big blobby thing in the background is that big fusion protein that I told you about. That's the problem. And then these colourful ones are the cyclic peptides that we're designing. And you can see they're fitting into the protein there, and that means it's going to stop that protein from being active. So here's just a little bit more data, and I will explain it to you. So down the bottom here, this was the results from testing some of those new cyclic peptides. And all it's showing is that here's imatinib, that commercial drug, and it's just showing that all these other cyclic peptides that we were looking at, some of them were able to be just as effective as the commercial drug. And this graph here, all it's showing is that some of these cyclic peptides, these cyclotides, were also effective at good concentration. So we don't want people to have to drink a litre of this stuff to get the effect. Hopefully they can just take a little thing. So what I've got up the top here is called mass spec, and all that means is that, so I actually took some extracts from my plants where I'm trying to make the best chronic myeloid leukemia drug. And I take the extract from the plant, shoot some lasers at it, all the particles break down and they fly at a detector, and then it comes out something like this. And this just shows me that there's something in that mixture that is the same mass as what I'm looking for. And for those of you who were paying attention before, when I said that all our cyclotides have 6 cysteines, I can actually also show that by doing a bit of chemistry and showing that this has moved 6 points over, and that means it has 6 cysteines in it. And so it's not just something else that happens to be the same mass as the thing I was looking for. It is actually what I was looking for. Another example is a Plasmon Inhibitor that we made in our tobacco relative. So a Plasmon Inhibitor is it's a molecule that was designed by some people in my lab to help with blood clotting. So often we want to get rid of blood clots, but in the case of surgery and after a traumatic injury, actually we want to stop blood loss. So there was a small molecule, a peptide, developed in our lab to treat this blood clotting, to encourage it. And that was published a few years ago, and then we actually that was made in the lab, and we want to make it in plants, and my colleagues were able to do that. So all this is showing is something that we produced in plants was exactly the same as something synthetic that was made in the lab. So these two things look the same. These also look the same, which means they're the same structure. And all this is showing is that the actual activity of inhibiting the plasmin, which is involved in the blood clotting, was the same as the synthetic version. So we were able to make the exact same thing that can treat this blood loss. We were able to make it in plants. So that was really exciting. And the last example that I'm going to give is an example of an anti-obesity drug based on a hormone that is from your brain that controls appetite. So we took 4 amino acids, a very small peptide, put those into one of our cyclic peptide scaffolds. And then this cyclic peptide scaffold looks a bit different. It only has 2 cysteines, and this is the one that comes from the sunflower. So we redesigned it, and we found that there were some good results from that. And this kind of idea of obesity sort of makes us think about perhaps, or leads into the idea of edible medicines. So what if you could have edible medicine like potato chips that could help you become less obese? It sounds kind of funny, but who knows? So a lot of the current plant-made pharmaceuticals from plants, they are extracts from a plant, which means they've grown in the plant, they've purified it in the lab. So there's still that laboratory-based component. But if we could have an edible potato or tomato or something with the medicine inside it, then we don't have to purify it anymore, and that can reduce the cost. If we have it in something like a seed, that also means that we can just store it on the shelf, and it doesn't require that cold storage, which comes back to the idea of helping people in countries that perhaps don't have very good access to medicine and don't have the facilities to store lots of medicine in refrigeration for a long time. So I leave you with these pictures of what you would like to take from it. Perhaps fries that can help you lose weight, or perhaps some seeds that might contain medicine you can sprinkle on your breakfast, or even a tea that you could brew. Or even, some leaves that have been freeze-dried and then put into a capsule. So what we're trying to avoid here also is this idea that some medicines, you do need to take them in other ways, i.e. having an injection. And as you can see here, none of us love injections, do we? So this guy thinks it's pretty terrifying. They all think it's pretty funny, but it's not that funny when you're next. So funnily enough, cyclotides actually do occur in some of some things that we ingest. So David was on a plane, I believe this is the story, was on a plane and got served this drink. And he thought, oh wow, that looks really colourful, I'm going to look up what it is. And it turns out it's made with an extract from this plant. This is that butterfly pea. And so it's actually used as a natural food colouring. There aren't many bright blue food colourings out there from natural sources. And so there's also a gin, a blue gin that's made here in Australia, and the blue colour also comes from this butterfly pea. It's actually really fun because if you have this gin, we had to buy some for our lab for analysis. It's actually really interesting because as you change the pH, so you might add a little bit of lime, it actually changes from blue to pink. They're not paying me to advertise this. But so we thought, since butterfly pea can make cyclotides all throughout the plant, as I mentioned in the shoots and the roots and the flowers, we thought, are there any cyclotides in here that might have come through the production process? So cyclotides are not making this thing blue, but it actually turns out that they do get carried through when this gin's being made. So there are some cyclotides in this gin. So there you go. Yet more evidence that cyclotides are stable, so they survive the gin making process, and we can ingest them safely. And so none of this would be possible if we couldn't discover these cyclotides in the first place. So I'm just going to have one or two slides at the end to show you some fun pictures of some of our discovery efforts. We're always looking for new cyclotides from plants. And last year I was lucky enough to be able to go on a trip to Western Australia, to the Kimberley region, to look for some plants that might contain cyclotides or even other interesting peptides. So we were looking for cyclotides from these plant families, and we found some really beautiful things out there. We found some of the butterfly pea. We found some native cotton. We also found some of the relatives of that plant from Africa that contained the cyclotides from the very beginning. They don't look all that interesting, so spot the medicinal plant in there, but it's in there. But we had a lot of fun collecting those plants. Here's just a picture of us putting all the samples into the sample packets at the back of the car. We were out for 10 days in the desert with no phone reception, no nothing, with our collection material. We were driving through the Kimberleys and just stopping everywhere we found a little bit of water, because we know the plants that we're looking for grow near water. So any little puddle like this on the side of the road, we're stopping and bending down and looking in all these weeds, and a lot of people will drive past and stop and ask us if we're okay because, you know, have we broken down or something? What are we doing? These weird people looking in the grass. And we said, no, we're fine, keep going. But then, of course, we did get a flat tyre. Not just a flat tyre, like completely blown out. And of course, that was when there was no one around. But luckily, I was with several people and we were able to fix it. I was actually driving the car when that happened, and that was a little bit terrifying. But I managed to pull over and we sorted it out. We also encountered some non-plant life, including some crocodiles. But they were freshwater ones, so they weren't too dangerous. We didn't go in the water anywhere near the salt water. So, as I mentioned, we were looking near creeks, and we found a few interesting ones. We've got Same Creek, Mistake Creek. Did we come here by mistake? I don't know. That was before our car broke down. And also, some of you might have heard of Wolf Creek. So, Wolf Creek is out that way. It's just out of the Kimberley region. But we thought, we may as well take a detour and go there. Some of you might know Wolf Creek from the horror movie based on true events of kidnappings in the outback. The kidnappings didn't actually happen at Wolf Creek, but that's where the movie's set. So that did make me a little bit nervous. So I'm going out there with my PhD supervisors with no phone reception. And they only played one prank on me and pretended to leave me behind one time. Some other people know Wolf Creek as this beautiful place, actually, that has a huge big meteorite crater, and it's also a significant place for the Aboriginal people in that area. And for us, we're the nerdy plant scientists who just wanted to go there to look for this plant. This is this tobacco relative that we grow in our lab that I mentioned was Australian native plant, and it was actually first collected from Wolf Creek, so we went there to look for that. Unfortunately, we had missed the rains and everything had dried up and we didn't find any, but we still had fun. So just to sum up, hopefully I have been able to tell you a bit about why we think making medicines in plants is a good idea for us and for other people on this planet, what it might look like. I've told you about what proteins are and what peptides are, and that plants can make these really special cyclic peptides, and that we can use those, redesign them to make new medications for different diseases, and that we would like to make our new cyclotide drugs in plants because we would like to make them cheaper and we would like to make their production better for the environment. And so we have gone from discovering drugs in plants to making drugs in plants. And with that, I will just leave you with a little bit of footage from our trip to the Kimberleys. This is my supervisor, Ed, who brought his drone along, so we could sort of survey the area and see what it was looking like in terms of the plant life, and also to take some fun videos. And with that, I just need to give some special thanks to my PhD supervisors, David Craig, who is here tonight. I don't know if you want me to tell you that. Mark and Ed and Marilyn. This is a picture of me with 3 of my 4 supervisors, and I hope that they don't think that taking me on was a mistake. I also need to thank Annie, who is very, very talented and helps everyone in our lab in a lot of ways, but her one particular talent that I would like to highlight is making Pac-Man animations on PowerPoint, so I thank her for that slide in particular, and some of the other slides as well. And also for Leonie and Dominic for contacting me and asking me to come along and organising this event. And of course, here are some Twitter handles. That's me. It's my email address. We've got a Twitter for our whole research group, and also for the Institute for Molecular Bioscience. So thank you very much.
Thank you so much, Bronwyn. Fantastic talk, and I'm sure you have lots of questions. If you do, now is your chance. There's already some come through on Twitter. You can also write them down, and Dom and Leo are going to come down the aisles, just wave your questions in the air like you care, and we will grab those. In the meantime, next month we are, of course, back on the 9th of September, and we are talking about mapping coral reefs from outer space and the need to understand what is actually out there in the environment if we're going to protect it. And particularly underwater maps are very hard to do. If you're on the ground, it takes a lot of time, so what can we do from space? So make sure to check out that talk. Of course, sign up to our mailing list so that you can get informed about upcoming talks. And if you head to our Facebook page, Twitter, etc., the videos of all our past talks are up on YouTube, so make sure you see those. All right, so we have some questions coming in. If you'd like to join us back on stage. Oh, thank you. Lots of questions. Fantastic. Let me try and pick one out. I'll go to Twitter first of all. So first question, we're going to start technical. Andrew on Twitter asks, how effective is the method of inserting DNA by agrobacteria? Is it 1 in 10, 1 in 2, and how do you check for it?
Right, that's a really good question. So when we use the agrobacteria to deliver the gene into our plants, there are a couple of different ways we can do it. So when I use the Arabidopsis plant, that was the kind of lab rat plant, it's probably more like 1 in 1,000 times, which sounds like it's not very much, but actually when you could see that what 100 seeds looks like, it's not very much. So I can get thousands and thousands and thousands of seeds. I can plant them in a tray that's just this big, and then I can actually use a selection marker to choose which ones actually got the gene incorporated. When we use the tobacco relative, the Nicosia Benthamiana, that's very, very effective. So we let the bacteria infect those leaves. We wait 5 days. We collect all the leaf material, and we can detect the peptide in there. Yeah.
Right. So that's following on from that a little bit, we've got a couple of questions, David on Twitter and a couple here, asking how you go about actually extracting the peptides with sufficient purity from the plants. How do you extract it and purify it?
So I'm lucky that I work with a lot of chemists and they know a lot about extraction. So basically what we do is we make a solution and we grind up the leaves or the seeds or wherever we think the cyclotide is going to be. We grind them up very, 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 very fine. So we're actually trying to break open the cells. And then we put them in this solution and we let them shake around for a while. And then we get rid of all the bits of leaf and things that are left behind. And we look in that liquid, in that solution. We look for the cyclotides in there. And there are multiple steps we can do to keep making it more and more and more and more pure if we need to.
Great. We've got a question here about the practical aspects around preventing contaminations in air or groundwater. A couple here, I think sort of both directions. One, how do you make sure that things don't get into your plants? And others, how do you make sure plants don't get into other areas?
Yes. So that is one of the big talking points, I guess, about GM crops. Can you just grow them outside? Are they going to get pollinated and spread their seeds around and escape? So that's why the GM plant industry is very, very, very highly regulated. And as you can see, actually, a lot of the plant-made pharmaceuticals at the moment are actually being made in glass houses, in contained facilities. So that's one way to stop them from spreading. In terms of getting into groundwater and things like that, these compounds we work with at the moment, we are in a lab that nothing can come in and out of that lab. And so none of our compounds can escape. And if that was ever scaled up to commercial production, it would be regulated in a similar way.
Similar, I guess, all the same issues around genes jumping into other plants.
Yeah. Well, I come from Tasmania and we grow eucalypt trees down there. They're not transgenic, but it's just an example of, that some eucalypt species were brought in to grow for timber, and they crossbreeded with the local eucalypt species, and then the local eucalypt species became hybrids, and then the species that were there gradually were declining and people had to do something about that. So it's not just the GM industry that's regulated in that way, but it's basically any crop that you want to grow, you have to make sure it's not going to escape into the environment.
We had a BrisScience talk a few months ago about GM foods that touched on a lot of issues if you want to know more about that. Speaking of genes, what did the gene, the agrobacteria gene in sweet potatoes, did it play a functional role in why it survived?
I'm so glad you asked that question, because when I was reading about this the other day, I also had the same question, and I went and asked my supervisor, and I tried to look it up as well because I couldn't figure it out. Why does the plant need a gene from bacteria? And it's interesting because the genes, I think there were 5 or 6 genes that got transferred from the agrobacteria, and some of them were genes for plant hormones. So first of all, why did the bacteria have a gene for a plant hormone? And then why would the plant try and keep that gene if it already has its own? And actually, I think we just don't know the answer to that. But the interesting thing is that these genes were detected in sweet potato that was around the time that it was domesticated. So perhaps, this is not fact, so don't quote me on this, but perhaps they did have some kind of advantage for the plant, making the sweet potato, like the roots, making them bigger or something, and that's why they were selected when humans were first wanted to eat them. But I don't actually know the answer.
Oh, well, that's always good to have questions you don't know the answer to. If anyone out in BrisScience land knows the answer,
yes,
tweet us. We'd love to hear from you.
Yes.
Well, we're almost out of time. I'm going to ask one more question, which is, what slide did you use when you won the People's Choice Award at the 3-minute thesis competition?
Oh, that slide's not in this talk. I'm so sorry. Oh, you'll have to come and see my 3-minute thesis talk.
Lovely bit of cross-promotion there. Great. Look, on that note, thank you so much to Bronwyn. And of course, if Inc Gin is out there and wants to sponsor BrisScience, we'd love to hear from you as well. But please thank Bronwyn for a fantastic talk and join us for food and drink afterwards. Thank you so much.
Thank you. Oh, wow, thank you. Thanks so much.
Make sure you join us next month. Have a great night, everyone.